Oxygen from the lungs is carried in blood in two forms. One of them is dissolved form, but of the oxygen entering the body, only 1.5% is dissolved in blood plasma, while the remaining 98.5% of oxygen is bound to the iron-containing protein, called hemoglobin, in the red blood cells. Now this hemoglobin at its normal concentration increases the oxygen-carrying capacity of blood to about 70-fold. When this oxygen reaches the body tissues, it is utilized by the cells, and cells, during various processes they carry out, release carbon dioxide. This carbon dioxide then enters the tissue capillaries and is transported back to the lungs. Now, hemoglobin consists of gluten protein that has 4 polypeptide chains, 2 alpha chains, and 2 beta chains. Thus, normal adult hemoglobin is called alpha-2 beta-2. Now, each of these chains bind a ring-like heme group, and each heme group has an iron atom in its very center. So, there are 4 iron atoms in total. Now, each of these subunits can bind to 1 molecule of oxygen, so binding a maximum of 4 molecules of oxygen to each hemoglobin molecule. But the thing is that hemoglobin might not be always bound with 4 oxygens. When it is not bounded with any oxygen molecule, it is said to be 0% saturated, as known as deoxyhemoglobin or reduced hemoglobin. Or it can have just 1 oxygen bound to it, and that would make it 25% saturated. While, if it has 2 oxygens bound, it still has space to bound 2 more. So we can say that it is 50% saturated, whereas hemoglobin bound to 3 oxygens, still having the capacity of 1 more, is said to be 75% saturated. And lastly, if the hemoglobin has all 4 irons bound with the oxygens, we call it fully saturated or 100% saturated. The binding of the first oxygen molecule increases the affinity for the second oxygen molecule and so forth. The affinity for the 4th oxygen molecule is the highest. This is called positive cooperativity. So it is the change in affinity that is responsible for the loading of oxygen in the lungs. Now, the oxyhemoglobin dissociation curve is the curve that demonstrates the relationship between partial pressure of oxygen and the percentage saturation of hemoglobin with oxygen. Here, partial pressure of oxygen is plotted on x-axis and hemoglobin saturation on y-axis. As a result, it reveals S-shaped or a sigmoid-shaped curve that describes visually how oxygen binds to hemoglobin, and this sigmoidal shape is due to what we call positive cooperativity. Now, oxygen-hemoglobin curve is shifted either to the left or right in various conditions. Shift to left indicates acceptance of oxygen by hemoglobin or loading of hemoglobin, while shift to right indicates dissociation or release of oxygen from hemoglobin or unloading. You can memorize it by their initial letters, L for left and L for loading, indicating that loading will shift the curve to the left, while R for right and R for release. So release of oxygen from hemoglobin will shift the curve to the right. Now, the partial pressure at which 50% of the hemoglobin is saturated is known as P50. If P50 increases, like this, it moves to the right. So, there is a decrease in affinity of hemoglobin for oxygen, and more oxygen is needed to get the 50% saturation. Lower affinity causes oxygen to release more easily, and oxygen-hemoglobin dissociation curve is shifted to the right. Now, what are the factors that can cause the right shift? Well, increase in partial pressure of carbon dioxide and a decrease in pH do so. Our tissues, due to higher metabolic activity, have a high partial pressure of carbon dioxide and a low partial pressure of oxygen, causing the carbon dioxide to diffuse into the blood, increasing the partial pressure of carbon dioxide there, causing it to diffuse quickly into the red blood cells. Here, in the presence of carbonic anhydrase enzyme, carbon dioxide combines with water molecules to form carbonic acid, which then dissociates into hydrogen ion and bicarbonate ion. Increase in hydrogen ions will cause an acidic environment with the decrease in plasma pH. Not only this, but both carbon dioxide and hydrogen ions can bind to the hemoglobin at a different site than oxygen, but decrease hemoglobin's affinity for oxygen. This decrease in affinity of hemoglobin for oxygen due to the increase in the partial pressure of carbon dioxide and decreased blood pH is known as Bohr's effect. This lower affinity enhances the unloading of oxygen into tissues to meet the oxygen demand of the tissue, and this shifts the curve to the right. In exercising muscles, there is more production of lactic acid, hence decreasing pH. Also, skeletal muscles generate heat, causing a rise in temperature during exercise. Both of these causes unloading of oxygen to meet the oxygen demand of the exercising tissue, and this shifts the curve to the right. Now, 2,3-biphosphogyl, or 2,3-BPG, is a byproduct of carbohydrate metabolism in red blood cell. It increases usually at high altitude and while doing a muscular exercise and hypoxia. 2,3-BPG binds with beta chains of deoxyhemoglobin, decreasing its affinity for oxygen, hence causing unloading of oxygen and shifting the curve to the right. So, all of these are the factors that shift the oxygen-hemoglobin dissociation curve to the right. Now, on the flip side, opposite happens for the left shift of the curve. It occurs when the affinity of hemoglobin for oxygen is increased and the P50 is low. Other factors causing it are a decrease in hydrogen ion concentration and increased pH, a decrease in the partial pressure of carbon dioxide, decreased 2,3-BPG concentration, and decreased temperature. Another factor is the fetal hemoglobin. Fetal hemoglobin has 2 alpha and 2 gamma chains, rather than the beta chains that bind 2,3-DPG. So fetal hemoglobin does not bind the 2,3-DPG as strongly as does adult hemoglobin. This increases the affinity of fetal hemoglobin for oxygen, hence the dissociation curve is shifted to the left. Now lastly, inhalation of carbon monoxide can result in severe poisoning, because carbon monoxide occupies oxygen-binding sites on hemoglobin, forming carboxyhemoglobin, thus decreasing the oxygen content of blood. In addition, binding of carbon monoxide to hemoglobin increases the affinity of remaining sites for oxygen, so it decreases the unloading of oxygen, and the dissociation curve is shifted to the left. So, that's what oxyhemoglobin dissociation curve is, and the factors causing its left and right shift. I hope you have enjoyed the video. Keep following sqadia.com for more such brief, yet informative videos.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.